This Week in Microbiology is brought to you by the American Society for Microbiology at microbeworld.org/TWiM. This Week in Microbiology, episode number 34, recorded on May 31st, 2012. Hi, everybody. I'm Vincent Racaniello, and you're listening to TWiM, the podcast that explores unseen life on Earth. Joining me today from Small Things Considered, Elio Schaechter.
Howdy. How are you?
Very well out here. It feels like summer. It's really hot.
Oh, good. You can use it.
Well, when it gets hot, then the power gets consumed and everything gets out of whack. But otherwise, it's beautiful.
A little hot is good. Too hot is not good.
Today we have some puffy clouds in the sky, and I understand that today's topic will visit why those clouds are there.
Well, maybe tangentially.
Tangentially,
at least in the southern hemisphere.
So we haven't heard you for a couple of weeks,
Right.
So good to have you back.
Thank you.
I hope you're all excited to.
Miss you guys. You went immunologically on me last time, which I think is fine once in a while, as long as you don't overdo it. That's fine.
Well, the immunologist would say you can never overdo it.
I know, but that's their problem.
I thought this that was an interesting union of bacteria in immunology. That's why we did that. If there were no bacterial connection, we wouldn't do it at all. Also joining us today, deep in the South, the Medical University of South Carolina, Michael Schmidt.
We're through hurricane number 2 of the year already, and it's only May, or tropical depression. But if they give the storms a name, I consider it a hurricane.
Well, really, I didn't even know that. I'm not paying attention.
Yeah, we're through Beryl. And the next one is, I have no idea, but I hope we don't see its name. Wow.
So you had a lot of rain and wind?
No, we didn't have we had rain, but not much wind. It was a weak depression, but the Gulf Stream is pretty warm, and the ocean is already quite warm. So
that means August and September are going to be tough for you, right?
Spooky, wow. Spooky.
Well, I hope that your samples don't get harmed.
Oh yes, the adventures.
So today we are talking about something different, I would say. Not the usual bacteria that we talk about, but rather another kind of microbe. After all, This Week in Microbiology, so we talk about all microbes. And these are protists. In particular, we're going to talk about one called Emiliania huxleyi. And this is a protist, right?
Well,
Elio?
wait a minute. Let me sort of amplify that. Let me tell you my take on this. The microscopic beings, which are not bacteria,
okay,
are huge in number and huge on the tree of life. If you look at the tree of life with the 3 domains and look at the eukaryote branch, at the very, very tip, you find animals, plants, and fungi. All the rest, all the way down, is mainly things which you cannot see with the naked eye. And so let's call them in general eukaryotic microbes. There's even a journal called Eukaryotic Microbe, I think. So that's legitimate.
Eukaryotic microbes. Okay, so we shouldn't call them protists. Protist is wrong?
Well, protist is one of them. Protist is one of them, and so are the fungi, and so are the algae.
Okay.
Now, notice that the definition breaks down a little bit because some fungi, of course, make mushrooms, which are visible with the naked eye, thank God. and some algae are huge, like kelp. So it's not a precise definition. In fact, these terms, as we're going to use them today, have no taxonomic meaning. This is not science in the taxonomic term. These are just common, ordinary words of the English language. So we're going to talk mainly about an alga, actually, a microscopic alga called Emiliania huxleyi. I'll talk about that later. But first, I'd like to introduce the whole subject a little bit because I'm a little bit of a fanatic about this. It seems to me that microbiologists tend to neglect this huge, huge aspect of life, and so do other people. Although they are of tremendous importance, they're of importance in the cycles of matter in nature. We'll talk about that later. They're important as pathogens. And they are now becoming increasingly important as possible sources of fuel, namely the algae. So here we have a group of organisms that has an immense importance in our lives. And we don't seem to be quite as up on it as usually in a microbiology textbook, such as, I gotta confess, I've written, these guys get a chapter. And in reality, they should get much more. So, with that in mind, let me first start out. I gave a lecture about this recently, so there is a big danger that I'm going to turn very didactic on you. So please stop.
Oh God, we're in for a treat, Vincent.
Yeah.
No, we're in.
I never heard Elio lecture. Yeah?
Right.
Have you, Michael?
Once.
Okay.
When we had
that was enough. That was enough. When was that, Michael?
When we had that meeting out at San Diego.
Oh yes, yeah. Okay. Anyhow, let me say first of all that these guys were among the very first things to have been discovered by Leeuwenhoek, because when he used his little microscopes made of a single bead of glass and he looked at pond scum, just like we did in high school, he saw protozoa and protists, he saw algae, and described them in beautiful, beautiful detail. Now, let me make another point. This is a point that I can't wait to make, but it's really an aside. You look at the tree of life, and appropriately, you find three domains: the bacteria, the archaea, and the eukarya. They are thought of as being divided by a huge cleft. And in fact, in terms of phylogeny, that's exactly right. That's what they are. But now think a little bit about the world today. The world today, are these things three separate things? Well, they are phylogenetically, but they're not with regard to ecology. All these things interact all the time. Our bacteria and archaea are found in many common habitats. They talk to each other. Eukarya, especially eukaryotic microbes, talk to archaea, talk to bacteria all the time, especially to bacteria. So I call this the tyranny of phylogeny. The idea that phylogeny imposes all your thinking.
Well, you really bring up a really good point about the molecular chronometer that is used to describe the tree of life, which is principally 16S ribosomal RNA. Right. And protein synthesis is universal. And this chronometer functions because it works on non-essential components of the 16S RNA molecule that can be subject to genetic drift without causing a lethal event. And so consequently, it really doesn't account for what you just said. The interaction is sort of absent. And it sort of assumes that everything is evolving by itself and not in concert with its environment, with the interaction of other species. And so this is a really important point for folks to think about as this story evolves and considering the importance of this really neat, and it is an animal for what it's actually doing out there.
Well, that's right. And I think that it pays to understand that ecology is part of the conversation at any step in biology. I would like to add to what Dobzhansky said, nothing in biology makes sense except in the light of evolution. I would like to add, and ecology. Mm-hmm. Nothing in biology makes sense except in the light of evolution and ecology. That's my take on it. Anyhow, let's leave the philosophy aside, okay? These guys have been around for at least 1.5 billion years, so they have a lot of time to evolve, and evolve they did into phenomenal shapes. Some make houses. Among them are the foraminifera, the diatoms, the radiolaria. I'm tossing out words. I hope that our listeners know what I'm talking about. And if they don't, please go and Google them, because or Google image them, because they're going to see shapes and forms which are so extraordinarily beautiful that it really tickles the mind. Anyhow, some of these guys are small, some are big. Some foraminifera make nautilus-like shells. They look very much like a nautilus shell. But they're a single cell, made by a single cell. And some of these reach incredible proportions. They get up to 10 inches in diameter. done by a single cell. By the way, the fossil remains of these guys make for limestone, and the limestone used for carving the blocks of the pyramids of Egypt are made of foraminifera. So there we have it. Anyhow, let's go. I can go on with the diatoms. The diatoms are something totally unique, and I think deserving so much more attention than they get. After all, they are made of silica. They have shells made of silica. By the way, the radiolaria have shells made, you wouldn't believe this, of strontium sulfate.
Wow.
Either of you ever hear of strontium as part of biology? Well, there it is.
Oh, yes. When you look at strontium from the nuclear bomb tests in milk.
Right.
Elio, you mentioned that some of them are pathogenic. What's an example of that?
Okay. Well, pathogens are among the protists. There are many pathogens like malaria, trypanosomes, giardia, leishmania, you name it. In fact, I'd like Michael's opinion on this. When it comes to the pathogenic protists, a surprising number are insect-borne. This is an aside from what we're talking about, but it's an interesting one. Let's deal with it for a second. I mean, there are many bacteria, not that many bacteria, which are carried by insects. Borrelia comes to mind, a few others, and some fungi maybe. But among the protozoa, we have, as I say, we have malaria, we have giardia. No, giardia is not insect-borne, but we have trypanosomes and so forth. Why do you suppose that is, Michael? Do you have a thought about that?
I think it's because of the niche they occupy in the host, and it's principally a blood meal that it moves them. And one of the things we're going to talk about today is sex, and sex is very important, but it requires movement. And so I think if you're going to anthropomorphize things, you really can see the role of an insect in moving one genome from one location. And remember, these are microbes. So when you just move from one tree to another, the potential for genetic diversity is pretty significant. So I think that's why many of the pathogenic protists, like our friend Leishmania, that comes from a sand flea or sandfly from a bite is so prevalent.
Yeah, I think that's right. And in addition, I think they have also evolved to be kind of weaklings. In other words, they don't do well in the environment. You couldn't transmit malaria by inhalation or by ingestion because they're too weak to survive in the environment. They need to be inside the body of something, whether it's the human host or the vector. But I think this is an interesting subject, and I'm surprised to see how general this is for so many different eukaryotic microprotists. Okay, well, are we ready to sort of tackle the subject of the day?
Sure.
Are we?
Sure.
Okay, so the subject of the day is an alga called Emiliana huxleyi. Emiliani was an Italian protistologist, and Huxley was Huxley, the guy who was known as Darwin's bulldog. And they both looked at sediments in the sea and they saw these amazing shapes. Now, what I'm talking about is a thing which is about 4 or 5 micron across, perfectly round, and covered with plates. And the plates look like grommets, something like that. This is one time when I wish we were televising this because it's so beautifully visual. And Emiliana huxleyi is among the most beautiful shapes in biology. Anyhow, covering this 4 or 5 micron cell, round global cell, are these plates, and there are about a dozen or so of them, depending on the species and also on the conditions. And these plates are made of calcium carbonate. And so what's that about? Well, obviously, it's some protective device. Covering yourself with a bit of pieces of marble is not a bad idea for survival. And so they probably resist ingestion. Now, the thing to be said about E-hux, let's call him from now on. E-hux is present in amounts in the ocean. It's, by the way, I think exclusively marine. It's found in the oceans in blooms. The blooms are so gigantic that they are bigger than the size of Ireland. I mean, huge. 100,000 kilometers square. I mean, really, really big stuff. And they make the water look white. And it can be seen as white water when you sail through it, or you can see this from a satellite. And the reason is that these plates of EHOX, these calcium carbonate plates, are reflective. So it's like a bunch of mirrors. And this, of course, has consequences on the heating cycle of the Earth, because these blooms are enormous. They tend to occur in the tropics and the subtropics. You don't find them in the Arctic, for instance. And they happen with some regularity, sometimes every year, sometimes less so. And they're gigantic. Now, before we go into that, let me say a little bit more about EHOX and other coccolithophores. They are algae because they have a chloroplast and they carry out photosynthesis. Other than that, the amazing thing is the formation of these coccoliths. Coccoliths is the name for these armor plates that they're covered with. It seems hard to believe. These plates are made inside the cell. They're made in a body derived from the Golgi. They are excreted full-sized. Okay? They go full-size from the inside of a cell to the outside. Now, these are a micron across or something like that. Imagine spitting out a thing like that and not losing your bodily integrity in the process. Can you imagine that?
Do we understand that?
It's absolutely incredible. I mean, it's really remarkable if you consider the pressure differential that they have to undergo and move these gigantic plates out. You know, when you move to thinking about how man is going to go off and colonize space, you got to take a page, because these really look like spaceships. They really look like spaceships in terms of how they're structured, and it's straight out of science fiction, but this is science reality. And I don't think we really understand, aside from the Golgi is responsible for forming the vesicle, and I think it's a series of doors in which you begin to move the plate from one compartment to another. And that's the advantage of being eukaryotic, in the sense that they have these double-walled membrane compartments, unlike the poor bacteria that only have their one unit membrane to move the product of the cytoplasm into structure. Everyone's going to immediately jump on me and say, well, about gram negatives. Well, that membrane is really different than the unit membrane that surrounds the bacterial cytoplasm. And remember, that's effectively what the Golgi are doing. They have the equivalent of a unit membrane around these remarkable compartments. But the other thing to think about with these compartments is they're giving you a sense of scale of how this little machine is assembling these very complex structures.
Very regular, by the way. They're all identical, essentially, on one cell. Yeah, you're right. I think it's amazing. By the way, diatoms do something like that too, and their shell is also made in plates inside the cell. So I wouldn't you have thought that these things are made extracellularly? I mean, before I looked into it, I would have thought, gee, just make them on a template outside the cell and move them over and make another one. That's not how it works. But you're right, Michael. I think this is because of the complexities of the eukaryotic cell. Plus, of course, eukaryotic cells have energy to spare because they have a number of mitochondria and they can make a lot of energy, which is certainly required on a big scale for this, don't you think?
Yeah, especially this one, because it's sitting out in the full open ocean where you don't have to worry about sunlight.
Right.
And the other interesting thing, which we're going to eventually get to, is that the whole water depth issue, because as you go deeper in the ocean, and the niche of this really cool microbe is the light-filled ocean. And it'd be nice to get Margaret's perspective on this. If she's listening, maybe she'll write us a comment.
Margaret
McFall-Nye.
Absolutely.
Before you go on, I found a movie of the
yeah.
production of these plates inside of
Yeah,
a cell and being extruded. I will put a link to that. There is
a link to it because it's amazing. It's absolutely unbelievable. These guys are suddenly boom.
Then boom, they're out. Exactly. Boom. You're looking at them form, and then boom, they're outside. And you, this eHux, by the way, has its own homepage, which is really incredible. Okay, go ahead.
Okay, the other point I was going to make, I didn't say that. The blooms are so gigantic that when these guys die, they die, but the coccoliths don't. These plates don't. So they fall down to the ocean and they make mountains. The white cliffs of Dover, gigantic structures, are made of the fossil shells, or the remains, when I call them fossils, the remains of the coccolithophores. So they have a huge impact on the carbon cycle, and we'll talk about that a little bit, but we'll also mention the sulfur cycle when we come to it. Anyhow, this is a overview of protists and Emiliania huxleyi in a few minutes. So I hope I haven't gone overboard with this, but I can tell, if you can tell that I'm enthusiastic about it, is because by gum, I am.
So you mentioned that the shell makes it resistant to being eaten.
Well, yeah, that's the assumption. I mean, nobody really knows.
Because presumably these are food, they're in the food chain,
Of course, but you gotta eat a piece of marble to get to the inside. It's like eating a marble in which the goodies are on the inside, surrounded by, let's say, by very tough stuff. So it would be, obviously, as we'll talk about later, viruses know how to do that, but I would expect that other microbe eaters in the ocean have a hard time with them.
Yeah.
Get an indigestion. I mean, imagine the brick you make when you have ingested these guys.
But you don't have to take a Tums because you already have the calcium carbonate.
Yeah, right. Yeah. So Elio, one question: how do they reproduce? These are diploid organisms.
These are diploid organisms. They divide by binary fission.
As
they do, they make more coccoliths, and they go their merry way. This is the question becomes more sophisticated or more interested in the diatoms. You remember there's some diatoms that look like pill boxes, or petri dishes for that matter.
Mm-hmm.
Now, here's a little aside. This is really an aside, but it's a good one. When these guys reproduce, the bottom of the petri dish becomes the top of the next one. And the rule for making the shell halves is that you make them on the inside of the previous template. So the bottom of what was the bottom of a petri dish becomes the top, and within it you make another bottom, and then you flip over and so forth. The result is they get smaller and smaller and smaller. This is unique in biology, a division that leads to size reduction. Okay, so what happens when they reach a point where they're too small to survive? They pull out of their shell, go for sex, make gametes. The gametes they mate and make a zygote, and the zygote starts over, making a new diatom, now with a big set of plates. That's pretty cool. It's so unique. Anyhow, I don't think this is true for these guys. It may be true for some other organisms which are surrounded by a tough shell. I don't know about that. Oh, can I have another minute?
Yeah,
can I?
Sure.
Another? Okay. One other thing, okay? Ciliates, like paramecium, and dinoflagellates, which are the guys who make red tide, have an incredibly complex cytological structure. I mean, it's so complex that in the case of ciliates, you talk about a mouth, a digestive system, essentially, a bladder, an anus called the cytoproct. I mean, all this in a single individual cell. And get this, in the dinoflagellates, in some dinoflagellates, you find a structure called the ocelloid, OCELLOID. The ocelloid is the equivalent of an eye. It's like a vertebrate eye. It consists of a cornea-like surface, a chamber, a lens, and behind it something that looks like a retina. The retina is made of modified mitochondria.
Wow.
Now, that's wow. I mean, really, that's all in a single cell. And of course, this is a light capturing organ.
Yeah. There's a blog called The Ocelloid, isn't there?
Oh, yeah. It's my pal. My pal.
You wrote this little review article about protists together.
Yeah. With Diana Egli, is her name. Yeah. She's a Very nice young person who's now at Indiana University and knows more about protists than I think all but a handful of people on Earth.
Yeah, this is called the Protist Wonderland. Yes. Well, I'm visiting Indiana University next year. I have to talk to her.
Hey, look it up.
I have to look her up. Okay. Today's two papers actually involve viruses of E-hux. And, well, you know, these big blooms that Elio talked about, they eventually crash. And so for a long time, people have said, well, maybe a virus or viruses are involved in this. It would make sense that the virus infects the cells and destroyed them. So about 10 years ago, virus of E-hux was discovered, and it's called E-hux virus, or EHV. And it turns out it is part of a very large group of viruses that have very big DNA genomes. This is a virus with a double-stranded DNA genome, and it's packaged in an icosahedral shell. And there are viruses of different sorts that infect all different algae besides E-hux, and also other amoeba. A Mimi virus, for example, is an even bigger one that infects amoeba. So these are E-hux viruses, and the two papers today deal with them. Now, the interesting thing about, or one interesting aspect of the virus E-hux interaction is that these viruses kill the E-hux cells, but some of them become resistant to infection, and they do that by becoming haploid. So the normal diploid E-hux is susceptible to infection with these viruses, but it's been shown in a very nice paper, which we will link to, that infection induces the production of haploid forms, which are then resistant to infection.
By the way, isn't that a little bit, there is a paradox here, because the haploid form comes out of the shell.
Right.
And it's like an amoeba. It's a weakling.
That's right.
And it turns out to be the resistant one. And this guy who's encased, the diploid, which is encased in this incredible armor cover, is the sensitive one.
Yeah, that's very interesting.
To show you.
And those of you familiar with your Alice in Wonderland will remember that this is the Cheshire Cat strategy. And that's effectively one of the titles of one of the papers on this very subject is the Cheshire Cat from the famous Alice in Wonderland.
That's right. So the assumption is that in these blooms, you know, you have infection, and then some of the cells die. You get production of haploid forms, which are resistant, and then eventually they have to become diploid again. So they can fuse and make perhaps diploid forms with genetic variation, and so you go through these cycles. It's very interesting. No one really knows how these haploid forms or why they are resistant to infection. One assumption is that perhaps there's no receptor for the virus, or maybe there are inhibitors. It seems to be regulated at the level of virus attachment to the cell. So either it's a lack of a receptor or production of an inhibitor. That remains to be discovered. Really interesting.
By the way, we should also say that the outside of E-hux is not just calcium carbonate. There's polysaccharides there, and there's other things, probably some proteins. So there's room there for receptors.
Right. And in fact, in one of the papers that we'll link to, there is a photo. Actually, it's on your blog. You have a post about this Red Queen business, and you have a photograph of these coccolithophores with virus particles attached to them. You can see it right on the surface there. Yeah. So these viruses are getting in, and they're quite big compared with the E-hux itself. So I'll tell you about one paper that was published in Science in last year, July 2011, and this is entitled 7,000 Years of E-hux Viruses in the Black Sea. So this is a very short paper. It's less than 2 pages. The idea here is, can we look in sediments, and when people do paleoecology, they take sediments and they go back many, many years, and they try and study what's going on in these various layers. And the idea here is, can we do this with E-hux and its virus? Can we look at changing host and virus populations over thousands and thousands of years that are represented in these sediments? And the Black Sea is a nice place to study because it's undergone lots of changes over the years in terms of its ecology and salinity, as it's very, very saline, and then fresh water has come in at various times. And this has been studied in a variety of ways. So in this paper, what they did is they have these sediments which go from the present back to 7,000 years ago. And since we know the genome of E. hux and we know the genome of the virus, it's a really relatively straightforward matter to make primers and do PCR and ask what's present in these sediments. So they did this, and they have basically one figure where they plot the genotype of E. hux, which is divided into 11 different genotypes, and they look across 7,000 years. And then they look also at the virus. And they're doing this by looking at specific viral gene and a specific E. hux gene. And you can see, for example, graphically, it's presented very nice. You can see they assign each genotype of E. hux a different color. You can see how some are present for a while, and then they go away, then they come back again, then they're replaced entirely by others. It's really quite interesting.
And they did it not via the brute force method of DNA sequencing, but they started out much simpler using a methodology called density gradient gel electrophoresis. And if you're not familiar with that technique, it's really pretty powerful. But to illustrate it...
You mean denaturing, John?
I'm sorry, denaturing...
Gradient
gel electrophoresis. Thank you, Elio. It's really pretty neat technology. It's the equivalent of the three of us were in an airplane, and we all jump out of the airplane at the same time with parachute. And since I am the least brave amongst these three, I will pull the ripcord on my parachute first, and of course I will be the last one to hit the ground. Elio, being the most brave of the three of us, won't pull his parachute until he's about 1,000 feet from the ground, so he will reach the ground first. And Vincent is in the middle, and he'll pull his parachute about halfway down. And it's the way DGGE works is it's based on the AT ratio of the particular PCR product that you have. And the way it works is they clamp at the front of the PCR primer, they put about a 40 base pair GC clamp. And this GC clamp remains together, holding the PCR product together, and then based on the AT richness of the genome in the denaturing component of the gel, and they have various concentrations of urea in the gel matrix, the DNA melts, and it's the equivalent of the parachute opening at the beginning, the middle, or the end of the jump.
See, it's a nice analogy.
It's a good way to quickly look at a lot of samples, and we'll see in the other paper that we do, if the DNA species migrate at the same place, they typically have the same sequence. And it's when they're different, then they have different sequences. So you can rapidly look at a lot of samples this way, and then if you want, you can sequence them to get more information. So they do that to look at the E. hux genome and as well as the virus. And over these 7,000 years, they've divided them into three ecological phases. The oldest, where the water was somewhere between fresh and brackish, and then there was a period of salty conditions, and then more recently, the last few thousand years, fresh water has been coming into the lake, and so they call this a freshening period. And you can see how the host E. hux is changing over these periods, and then when you have in the oldest sediments, the viral diversity is rather constant over the years. There's some changes, but then in the most recent time, the last 3,000 years, where you have freshening of the lake, you have a much greater diversity of viral types. Mm-hmm. So it's a really nice way of looking historically at historical samples like these and try and make correlations between the host and the virus, and of course, in whatever ecological conditions have occurred, so you can make correlations between them.
By the way, I had to readjust my thinking here when looking at this because the abscissa is calendar years before the present, and so zero time is now.
Yes.
You're
Yes.
used to thinking about zero time is going to be at the other end.
Yes, that's right.
So anyhow, it's cute.
Yeah. So he says in the end, this provides insights into long-term virus phytoplankton host dynamics during the Holocene. So it could be useful to do this. So not only can you look at other parameters that these sediments are good for, but now you can look at viruses and hosts specifically. So that's the power here. And they used E. huxleyi to do this because it's in sediments, right? That's what they're looking at.
It's sort of surprising that the virus stays around as long as it does. I mean, the EHUX I can see because of its anatomy, but these viruses are pretty tough.
Yeah, I think that's...
they may not be viable, but they're present chemically at least.
Part of the... that's the point here. We don't know if these are infectious viruses because they're in
someone once told me, and it was a phage biologist, that the most stable form of DNA that's ever been devised is the form of DNA that's encased in a phage coat. The DNA is properly dewatered and stabilized inside the phage head, and it remains the most metastable form of DNA that's ever been constructed. So if you want to preserve your DNA, get it packaged into a phage, and you can leave it sit out on the desktop. And as long as you don't have a gamma source and ultraviolet source hanging around over the test tube, it'll remain stable. I think, I'm trying to remember who it was. It was someone in Texas, and the name will come to me in a minute.
Anyhow, that's a very interesting datum, and I'm glad to hear it. I didn't know that, Michael, so thank you.
So the last thing we'll point out here is that what the author concludes, historical plankton virus populations can be included in paleological and paleo-environmental studies. So that's the strength here. So there you go.
By the way, there is another paleological fact which is interesting. The coccolithophores typically make compounds called alkenones, which are C37 to C39 carbons long, and they are extraordinarily stable, just like some steroids are extraordinarily stable. So you can do chemopaleontology with some of these things. Right. That's not a virus, but that's at least the host has a chemical signature that can be seen in very, very old sediments. I think that's pretty neat.
Then my neurons fired. The individual's name is Dr. Rai Young from Texas A&M.
Oh, yes. Yeah, he would know. Of course.
Okay, our other paper is similar. It has to do with the composition of E. hux and the virus in a specific bloom that occurred in the North Sea in 1999. And Elio will tell us about that.
Yeah. Well, it's about looking at the dynamics. Well, let me read the title. It's Dynamics and Genotypic Composition of Emiliania, huxleyi, and their co-occurring viruses during a coccolithophore bloom in the North Sea, and it's published in FEMS Microbial Ecology of March 8, 2012. By the way, the date has some significance because the samples were collected in 1999. So a student asked me, what gives? Why do they wait this long to publish in 13 years? And it could be any of a number of reasons. They may not have had the budget for it. They may have had other priorities. I don't know, but the samples are stable, and so you can do research on something that you collected many years before. Who knows? I don't know, but it's a cute point. Anyhow, so what they did here is to go on an expedition, and this expedition bears saying what this was all about. It's called the dimethyl sulfide biogeochemistry within a coccolithophore bloom, and this is abbreviated to DISCO, and it's in the northern North Sea, June 1999. We need a digression here about dimethyl sulfide, don't we?
Sure. Yeah.
Okay.
Yeah.
All right. So here's a story. It has little to do with what we've been talking about today, but it has a great deal to do with the weather. Now, you heard me right. I didn't say climate. I said weather. The difference, climate is what's in general, and weather is what's tomorrow or yesterday. So we know that microbes have a great deal to do with the weather because they participate in a big way in the production of atmospheric gases like all oxygen is originally thought to have been derived from cyanobacteria. And that has enormous consequences on the climate. That is, over the years, as gases change in composition, the part of it is the greenhouse effect and other things. All of this has to do, climate has to do with microbes. There's a little question of that. But what about the weather? The weather is, as I say, the clouds here that Vincent is looking out from his window in New York City. Well, here's the story. Emiliania, huxleyi, and other algae make a protectant, an osmoprotectant, a compound that protects them against the high osmotic pressure in the ocean, as well as radiation, called dimethylsulfonylpropionate. It's a simple chemical, and it is excreted in enormous amounts. I mean, this is really tons and tons and tons. Millions of tons of this is made every day. So what happens is bacteria, especially of a group called the Rosiobacter, which is a very important group of marine bacteria being studied in a variety of places, including in Mary Moran's lab in Atlanta. These bacteria take DMSP and make it into DMS, dimethyl sulfate. The methyl sulfide is a compound that we all use in one connection or another. It's a nice smelly thing. It's the reason why when you go to the ocean side, you have these wafts of beautiful ocean breezes turn out to be not very different from what you could smell in your toilet at less cost. Because we make the... Anyhow, dimethyl sulfide is volatile. DMSP is not, but dimethyl sulfide is volatile. So it goes up into the atmosphere, and into the atmosphere, radiation converts it into different sulfur-containing compounds, including sulfuric acid and others. And these act as nucleation centers for water droplets. In other words, they make clouds. Okay? So now this is a cycle, and it has an interesting feedback loop, which Lovelock, who proposed the Gaia hypothesis, was very quick to latch on. Namely, now you make clouds, you have less sunlight on the water, you have less algal growth, you have less algal metabolism, therefore less DMSP and less DMS. But you make less of that, the clouds go away. So it's a cycle where there is essentially a possibility for a feedback loop. Now, before we get too carried away, let me point out the truth that you probably all know, that a tremendous amount of sulfur which is produced, which is put in the atmosphere, is done by people. In power plants, in many of the things we burn, there's a lot of sulfur compounds that go up into the atmosphere. So it's not, what I've read, and I don't know exactly where this stands right now, is that this statement that microbes are connected with the weather is probably more true in the southern hemisphere, where there are very much fewer anthropogenic forces. There's less industry, let's say, than in the northern hemisphere. But important it is. And so people are looking at these guys, since EHOX is a major player, maybe the major player, looking at these things in connection not just with the sulfur cycle and the carbon cycle. They are part of what we know about. And I should also say that the reflection of sunlight by the coccoliths, by the covering things, the scales covering the coccolithophores, is also something that's being considered. And so all of this is part of global warming and the concerns we have today. Okay, that was that digression.
Well, let me ask one question. You made a really intriguing statement. If this is true, could these blooms then be responsible for things like drought?
Well, yes and no. That is. The literature that I haven't, to be honest, I haven't read this in any great detail, but there are contradictory statements about whether the coccolithophores are a sink of carbon dioxide or whether they produce carbon dioxide. So the jury is out. I think it's the best.
Yeah.
I don't think we know, but I know that very serious people look at it and they send whole ships to study this, and there's really, tune in, because there'll be much more said about this. It's just that I'm not willing to stick my neck out right now.
Well, that's why this ship went to this bloom, right? To do exactly that, right?
That's fine. Anyhow, so they went and they collected a bunch of 1 liter samples and they filtered them on board and froze the filtrate, which was consisted supposedly of the particulate matter, namely algae, and their viruses. They collected on that, and then they did pretty much what was done on the other paper. They looked specifically, however, for a calcium binding protein gene in EHOX, and they looked at the major capsid gene for the EHOX viruses. And what they found is somewhat predictable, I would say. They found that during blooms there is changes in both the space where these things are found and in the sequence. What happens when a bloom crashes? When a bloom crashes, these guys, most of the coccolithophores are dead. Some go haploid, and slowly they rebuild a new population. However, it turns out that non-native viruses participate in this. So there is an influx of viruses from a different population of coccolithophores. And this is it's sort of an interesting proposition. In a way, the way to think of this is a little bit like in some bacteria and some protozoa show antigenic variation. That's a way in which they defend themselves in the body of their host against a specific antibody, let's say. They just simply vary, and so they go from one to the other. That is what recurrent fever is like, brucellosis is like. A bunch of diseases show this. So in the ocean, EHOX goes through transitions such that it's no longer sensitive to one kind of a virus. It goes on and becomes sensitive to a different one. And of course, this is sort of almost like the Locke-Volterra mathematics, the idea that predators and prey alternate in their frequency. This is pretty much what you see here, but it's a little bit more complicated because the EHOX changes in location in the ocean, and they tend in time to sink, where they do this even before a bloom. So you find a variation in time between the presence of the depth at which you find EHOX. Now, they think that you can see this as the red queen hypothesis to alternate it with what we talked about before. The red queen hypothesis says that the red queen is running in place in order to stay put. So it takes changing all the time. This is also known as the kill the leader hypothesis. And this is the opposite of the Cheshire cat mechanism, which is instead of
hiding in plain sight. The Cheshire cat is hiding in plain sight.
It's becoming haploid instead of being diploid would be a case for the Cheshire. So it is possible that we see here both mechanisms happening at the same time. And the general conclusion is that this is complicated stuff, that this is not as simple as tigers or lions and zebras, but there is really much more to it, and there are many, many factors which are involved, including the location and the time of year and all these things. So it's a nice general conclusion. The specifics, I think, need a tremendous amount of work to be worked out still.
They look from June 18th to the 29th, and as you said, at different depths. And it's interesting, at one point they say there was an influx of warm water into the bloom.
Yeah,
and you can see that correlates with a change in the host as well as the virus.
Right.
So you're bringing in new cells and new viruses with this warm water. It's really interesting, but it is all correlative, right?
Yeah, that's all it is. Sure. I think they could probably do these mesocosm experiments. Marine biologists do this. They take a chunk of seawater that can be a liter, 10 liters, 100 gallons, whatever, or much more, and or even in the ocean itself, they put a barrier between the site. And in such cases, you can do a lot of studies under more controlled conditions because you can add something to it.
Exactly.
And I don't know how much. I think some of this has been done, but I'm not really aware of it.
So let me ask you a question about mesocosm. Is the standard microbiologist mesocosm, a TSA blood agar plate that we routinely use to plant the bacteria we recover from a human host in order to figure out what's growing in a lab, or Mueller-Hinton agar. And is that a right mesocosm to begin to characterize what's going on? Well, as I was reading this, I was trying to think about parallels that folks could relate to in bacteria. And the one that immediately came to mind was the story about how scarlet fever, which, if I'm remembering right, is the toxin is mediated by a phage, and the phage waxes and wanes in the population, as does the expression of the toxin, and as has scarlet fever had blooms in populations over time. And so I think what we see in this very elegant 7,000-year story with this EHUX that goes through a haploid state in which you see the expression of a phenotype. And these guys were very clever in using a molecular chronometer that's actually tied to the biology of the microbe that they're studying. They're able to begin to decipher this complex dynamic that's occurring in real time based on location and depth and weather. And this is what really the human condition that we call virulence or pathogenesis is all about. So I think our friends in pathogenic microbiology could take a page by looking at some of these really neat papers and begin to think about how to adapt them to their own studies.
Yeah, I think you're right. I think this is all, these are become models on how to do certain studies. I think in both cases we can say that. Yeah.
Now, the original purpose of this, remember, was to look at, it was called DISCO, dimethyl sulfide biogeochemistry within a bloom. And they say in the discussion, they really didn't see any effect on DMSP production during the course of this study.
Right.
You know, so even though the cells are dying and replaced and the viruses are changing, the DMSP is still the same, right?
Yeah.
Well, it's probably doing what it was physiologically intended to do. And what they were measuring is a protein that's critical to the diploid component of the animal that they're studying. It was a calcium.
Don't call it animal. Don't call it animal.
All right. You carry on. You carry on. It's they're using a protein that is specifically tied to calcium binding. We'll be very specific.
Right.
And the question is, did they try? Because oftentimes negative results never get published. So did they try a set of PCR primers that will specifically go after DMSP? I didn't have time to dig into the literature to find out if that's already been published.
Yeah, I don't think so. I think they just looked, for the chemical.
Yeah. They weasel a little bit. I mean, they say, well, different strains of EHUX make different amounts of DMSP. They are the influx of warm water, blah, blah, blah. I mean, there's a lot of... They don't like their own conclusion, clearly.
Yeah,
they would have liked a different effect, and I mean a bigger effect on the MSP and the MS production. But they don't think the last word is in fact.
Well, of course. I mean, I think the virus killing obviously has some role here.
Right.
It may not be on DMSP production, but it could be in turnover of carbon or sulfur. Who knows, right? I think it remains to be done, yeah.
Right. Well, I would suggest to all of us, and our readers included, that they pay a lot of attention to this.
Yeah, it's very cool.
It's really important stuff.
Okay, we do actually have a couple of emails today, so let's go through them. The first one is from Peter, who writes, I see that some action is now being taken in America against the non-therapeutic use of antibiotics as livestock growth promoters. And he links to a couple of articles. One of them in the New York Times and another at the FDA. Having read the FDA press announcement, I think that the New York Times article may be misleadingly optimistic. I feel that this is probably too little and too late. The practice of using antibiotics as growth promoters was stopped in Sweden in 1986, the UK in 1988, and in Europe by 2005 without significant adverse effects on the meat industry. As I see it, the only reason this practice persists in America is the lobbying by the livestock industry against the scientific consensus. Your view, please, doctors.
Mm-hmm.
Well, I just heard a talk by Barry Eisenstein, who gave a sort of
Who's
an
a
overview.
distinguished developer. He's a distinguished developer of antibiotics, by the way.
Yes, he is the first one to publish with Fred Starling out of UNC that Neisseria could have sex with E. coli and transfer the beta-lactamase gene, vice versa. Yeah. And that was published in Science, and he and Stanley Falco, and then Falco published, Eisenstein published in Science, and Falco published in Nature. And, that was when the gonococci were becoming resistant to the penicillin derivatives. And he gave an overview. He is now vice president of Cubist for science policy. And he gave a fascinating overview. But one of his opening slides was antibiotics have been with us almost as long as we've had bacteria. The bacteria, of course, are off. The bacteria and the other microbes, like fungi, are responsible for these antibiotics, and they've been around for millions and millions of years. And so the microbes have learned to adapt quickly to them. And we've only had about 60 years of antibiotics, and there are precious few targets left that we haven't developed. And he gave a very sobering talk to the infectious disease residents about the future of drugs. And he really pointed out how if we don't take antibiotic stewardship seriously, all modern medicine will stop. Because if you think about it, you can't do hip replacement without antibiotics. You can't do cardiac surgery without antibiotics. Can't do a lot of things in modern medicine.
You kill the patient.
Yeah. You can do them, but it's bad news. And so I think our person writing the letter is really taking the tone. Is it's a serious wake up call, and we have to begin to get serious about developing new antibiotics. We have to think about how the pharmaceutical market is going to be able to bring an antibiotic to market for the ethical use in people. And the trivial use of reducing the farm to table time, which is really an economic consideration. Are we willing to pay more for the protein we eat so that we can live longer?
Well, it remains to be seen. It remains to be seen if it's even at a cost. Because what the caller is saying, what Peter is saying, is that in Europe, and I think it's right, this was not at the detriment of meat production.
Yeah.
So it may not be that it costs.
Yeah, they're just arguing farm to table time, but there's no data to support it.
Right.
So the point here is that this is a voluntary action that the FDA is called for. In other words, it's not required. You're not required to eliminate the antibiotic. So is this going to be effective in the U.S.? That's the question.
Right.
And I think it's a good point.
Well, we saw how the general public was able to get rid of that meat byproduct that they were sneaking into hamburger. It only took ABC News, a couple of stories on mainstream television, and immediately all the major vendors of ground beef immediately, what was it called? Pink slime, got rid of the pink slime in the beef. And so I think this is an issue where if we really begin to understand the seriousness of the problem, that without stewardship of antibiotics, we're going to see medicine regress, then it's really going to the general public will wake up and demand action, and the meat industry will get rid of it. Will go the way of pink slime.
So what you're saying is it has to get a lot more serious before anything will happen.
Yeah.
Yeah. Okay. So there you go, Peter. You're right. And I agree. I think that it has to get very dire before, because the microbiologists have been hammering on this for a long time, as we heard at GM and ICAC last year. The message has been passed for years, but nothing happens. So I think that it's got to be a stronger action than the FDA saying you should do this. They should say you have to do it. So that's what we think, Peter. All right, the next one is from Sergio, who writes, I have a question for Michael Schmidt.
Oh, no.
Is there a Michael Schmidt here?
Yeah, it looks like my oral qualifying exam all over.
Well, doing my research on R. solanacearum, I don't know how to pronounce that guy.
Solanacea.
Okay, I read an old paper where they mentioned that pseudomonads were able to internalize copper and therefore detoxify a copper-rich liquid medium. I have observed this myself with the R. solanacearum strain I was working with, but since Michael is the copper expert on TWiM, I would like to know if there is some more information about this phenomenon. It is of my interest beyond my doctoral thesis because copper is one of the few fungicides allowed to be used in organic farming. Therefore, this phenomenon deserves more attention from organic farmers, I think.
Well, it's a really terrific question. And he is getting at the heart of how copper kills bacteria and how clever microbes like the pseudomonads get rid of it. One of the hallmarks, the way pseudomonads or Pseudomonas in general gets rid of toxic compounds, that it makes these tremendous pumps and pumps them out. And effectively, what the pseudomonads are detoxifying copper-rich liquid medium is they're probably moving the copper. And I don't know of a specific mechanism of how Solanacarium is doing it, but my suspicion is the concentration is below a critical threshold because you're dealing with ionic rather than metallic copper. So what you're effectively doing is dealing with a monovalent or divalent cation that's competing with one of its competitors. In the case of copper, as a divalent cation would be competing with the magnesium, and which will be a cofactor for many of the important enzymes. And in the case of the process that I study, we're actually using metallic copper, where it's a completely different mechanism of how the copper is effectively disrupting the normal physiology of the microbe and rendering it dead. In the case of fungicides, I think what they're doing is they're applying it, and again, I have to look up the specific form of copper in the fungicide, but my suspicion is it becomes ionic. Elio, do you know which type of copper is in the copper fungicide?
God, no idea.
Yeah, I think it may be a derivative of copper sulfate. I'm just going to do a quick Google and see what it's labeled as. It's just called shopping for copper fungicide. So I'll look it up and we'll get back to you.
All right.
It's copper sulfate. The active ingredient is copper sulfate. So that's what it is it's effectively serving as a competitive inhibitor for essential cations that are used for normal physiology.
All right. Sergio is from Bolivia.
Oh,
how about that?
Lovely country.
You've been
lovely
there?
Lovely country. Yeah. It's an amazing country. Really, the landscape is like nothing else.
Actually, I
the
think
people are wonderful.
I think that fellow who does the podcast Mundos de los Microbios, I think he's originally from Bolivia as well.
You see, I don't remember if he is, but
I think so.
Could be.
The next one is from Sarah, who writes, Dear TWiMologists, you know how sometimes you run across a word or phrase which sounds like it's got a specific meaning that's really important to the context of whatever you're reading or hearing? Well, I've been catching up with episodes 27 to 29 lately, and true to TWI form, the phrase type 3 secretion got the gears cranking. The phrase sounded significant, but I didn't remember learning about secretion mechanisms under this framework in undergrad 10 years ago. I did a little reading and wanted to share how fascinating these mechanisms are and how relevant they seem to be for many common human pathogens. Depending on the bug, the product, and the environment, these guys can deliver a variety of toxins into the extracellular environment, spit out antibiotics and other compounds, or deliver proteins or nucleic acid directly into target cells via injection or vesicles. What a spectacular array of intricate and elegant mechanisms, as Dr. Schmidt would say, remarkable.
Hey.
Are you getting a lot of attention this week, Michael?
I am. Well, at least we have listeners.
That's good, of course. Yeah. It's fitting that these podcasts are award-winning. They always nurture curiosity and prompt trains of thought into unexplored territory. The discussion about tunnel and apoptosis in episode 29 reminded me of a suggestion for a pick of the week, a couple of music videos that Roche released in 2009 to promote some new lab tools to measure viability and cytotoxicity. They're more for tissue culture than microbiology, but hilarious nonetheless. And so she provides some links to these videos by Roche, which we'll put in the show notes so you can take a look yourself. So thanks for that, Sarah. Thank you for writing. We appreciate it. And we are glad that we nurture your curiosity. The last email is from Stephen, who sends a link to an article in Night Science Journalism Tracker. And it is basically a review of TWiM 32, which was our chat with Rosie Redfield about the monolake bacteria. And the author of this review, who is Charlie Petit, he says that take a listen to four very savvy and plain-talking biologists chatting on their business at an Inside the Academy site called This Week in Microbiology. So he really liked our discussion. He says the conversation is excellent and another example of the myriad 21st century ways by which science information makes its way freely to the interested public.
Isn't that nice?
It's a very nice.
I must say, I remember that TWiM is a particularly satisfying one.
It's very good.
Really fun. Really good.
And Stephen, thank you for sending it. Stephen comments, horizontal transfer between blogs. It's very good, Stephen. We like that.
Very good.
So that is TWiM 34. We have all our show notes, by the way, at microbeworld.org/TWiM. You can also play the episode there, or you can get it on iTunes or the Zune Marketplace, wherever else you like to hear it, or you can listen on your smartphone with the Microbe World app. And as always, if you like TWiM, go over to iTunes and on its page, leave a comment or rate it. And that helps us to stay prominent so more people will discover us. And if you have a question or comment, we do like to get them. Send them to twim@twiv.tv. Elio Schaechter can be found at his wonderful blog, Small Things Considered.
Thank you.
Good to have you today, Elio.
It was a lot of fun.
Good.
I'm glad you let me talk about one of my favorite hobby horses.
Anytime. We love to hear you talk, and any kind of favorite thing you have, you're welcome to talk about.
I got a few.
Apparently, yeah. Michael Schmidt is at the Medical University of South Carolina. Thank you, Michael.
Thanks, Vincent. Again, this was an absolutely fascinating episode.
What's the you say fascinating? Is that what you said? Remarkable.
Remarkable, I think is what the question was.
Yeah, it's always good. I learned a lot this time because I didn't know much about EHUX.
Yeah.
And this virus connection is very interesting. So that was good. Thank you for stimulating us to do that, Elio.
Oh, my pleasure.
My name is Vincent Racaniello, and you can find me at my blog, which is at virology.ws. I'd like to thank the American Society for Microbiology for allowing us to do TWiM, particularly Communications Director Barbara Hyde, Chris Kondayan, and Ray Ortega for their technical help. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.